Welcome, everyone, and thank you for joining us for this unique opportunity to directly address awardees of the Research on Humans Division of the Brain Initiative. It's my pleasure to introduce our panel of experts who will be available to answer your questions during this live Q&A. Our experts are Dr. Elizabeth Buffalo, Dr. Ueli Rutishauser, and Dr. Robert Gaunt, who will be sitting in for Dr. Jennifer Collinger. For more information about our awardees, please refer to the speaker biographies in the center of your screen. My name is Dr. Roshan Akashamune, and I will be the moderator for today's Q&A session. LabRoots is delighted to bring you this incredible opportunity. We would also like to extend our special thanks to the Brain Initiative program directors, and in particular, Dr. James Nat, for their efforts in organizing today's session. This session is interactive, so please submit your questions by clicking on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. We will address as many questions as time allots for. So let's get started. Our first question is for Dr. Buffalo. Commonalities in the structure of stimuli promote our learning. Under what circumstances or how can our mental schema be upended? How is it? What are the powerful triggering variables?
Thanks. This is a really interesting question and something that we're hoping to address as part of our research into the U19 grant. As I was talking about in my presentation, it's these commonalities in the structure across trials or across stimulus modality that we think leads to the formation of a schema, which may support rapid formation of new memories. And we think, based on data that's been collected in previous studies in rodents, that the hippocampus can support this kind of schema development. And that we know that the response of hippocampal neurons stays constant through subtle changes in the context or the trial structure. But these responses can change dramatically as soon as the animal attends to something novel in the environment. That leads to a different kind of behavioral response. And so we think that really when the mental schema is upended, that happens due to really strong changes in the relationship between what the organism is doing and kind of the response or reward outcome.
That makes me think about, Dr. Rutishauser, your talk where you talked about cells that scale with familiarity. How would that speak to what we just heard as far as being able to upend, where you've got these neurons that are context-dependent responsive versus familiarity-dependent responsive? So how would that balance sort of play into this? That's my question, sorry.
Yes, thanks for that question. So first of all, thanks for having me. This is a very unique opportunity. It's the first time I'm doing a talk from my office, so bear with me as I get used to that. Yeah, so that question fits very perfectly. So in fact, what we have found is that there are some neurons, as you mentioned, which care about memory strength, meaning they care about how familiar something is. And the more they fire, the more familiar it is. But we also described another group of neurons which only care about whether you have seen a stimulus never before. We call those novelty neurons. And presumably those novelty neurons would be, if I understand Beth's argument correctly, would be what triggers the instantiation of a new schema. And what's really interesting about these novelty neurons is that they really only fire in terms of increasing their firing rate the very first time you see something. The second time, they don't react anymore. So they would be ideally suited to trigger this initiation of a new schema, I would think.
Well, we've seen those kinds of cells across different modalities, not just in the visual system as well. So it's that's interesting. We have a question for Dr. Collinger or Dr. Gaunt. The study and your findings have been quite interesting to learn about. If the study continues, what are some of the next hypotheses and tests you are considering to further advance the knowledge in the space?
Yeah, I'll echo everybody's comments so far about it. Thanks for being involved. Thanks for this opportunity. And it's certainly unique not have to fly somewhere and sit around in a hotel to do this. So it's quite nice. And I'll do my best to fill in for Jen on this. She's on family vacation this week. So I work with her closely on these projects. So again, I'll do my best. So to the question about what do we see going on in the future, first of all, we really do want to keep doing this sort of work. It's, more and more labs are starting to do this, and which is fantastic.
Can I interrupt you for one moment, please? Just for the rest of our audience, would you please describe a little bit the project?
Yeah, so the project we're working on really is focused on trying to restore dexterous motion in people who have lost it because of injury or disease. And so we work with people with degenerative disorders or spinal cord injury who can't move their arms anymore. And really what we want to be able to do is extract information from the brain to allow them to control a robot or some device, a computer, whatever they choose, as well as be able to put information back into the brain through stimulation of somatosensory regions of the brain so that they can perceive sensation. So you can imagine being paralyzed, reaching out with a robot arm to pick something up, and then feeling it as you contact it, as you grasp the object, as you grasp whatever it is you want to, and then bring it back to manipulate it. And so those are the sorts of experiments that are going on routinely in our lab right now. And the questions thinking into the future that we're particularly motivated by are ones that lead to better use and better ability to control these devices. And so the things that we can't do very well right now, I would say, are dexterous hand motions. And so while we can pick up a water bottle, pick up large objects, picking up fine instruments, pens, coins, things like that, those are difficult for us. And we think that sort of the integration of sensory feedback into the control will improve that. But those are learning how to extract information to be able to do dexterous hand control, as well as ways to be able to integrate sensory information back into that are the sorts of tasks that we're working on and thinking about in the future.
Thank you. We have another question for you, Dr. Rutishauser. Your recordings are from epilepsy patients. Can't memory be affected as part of morbidity? And how would you normalize for that?
Yes, thanks. Yeah, absolutely can. So, in fact, that's one of the main reasons we do this research is that we would like to figure out how we can address this comorbidity by developing new treatments for memory disorders. So epilepsy most definitely affects ability to memorize. So the way we deal with this is that we carefully characterize our patients in terms of their ability on standardized tests, and we see whether the mechanisms we describe are affected by these scores, and by and large they are not. And we also, while epilepsy on average does affect memory, for many people, in fact, it does not. So they have normal scores. So we can look at that subset alone and compare it with the set where it is affected. And one finds, by and large, the same mechanisms. However, there are interesting ways that are revealed by doing this, by which epilepsy does affect memory. And one of the things that we have described and that we have found is that these novelty neurons that I talked about before, they still exist. So if you compare a novelty neuron in the hemisphere which was later resected versus the hemisphere which was not resected, which was healthy in quotes versus not, you find that the novelty neurons in the later resected hemisphere, they still exist, but their response strength is weakened. So that's a potential way that you can explain why they have a memory deficit, in that their novelty detection mechanism is impaired in that sense.
I actually saw a related question to that here for you somewhere. Here. Would you say that confidence increases memory retrieval? Or, oops, sorry, was that it? Yes. Would you say that confidence increases memory retrieval or capacity, or is it just that these familiarity and novel neurons that scale with confidence are simply responsive to familiarity? And then that, I think, speaks into when you have the epilepsy patients. I'm not sure if you recorded how close to an event when they have an aura or not, and how that might also affect it.
Yes, those are two very interesting questions. So the first question, that we think that confidence, that the sense of confidence is a direct correlate of memory strength. So that means, and that correlate of memory strength is relative to the memory strength that that person has. So if a person has high confidence in a particular trial, they would have high memory strength relative to whatever memory strength they're able to form. So confidence is a very subjective judgment, which is relative to your own. Epilepsy patients we have tested this, they have very good insight into their memory strength. So it seems like their ability, this is a metacognitive judge, to make metacognitive judgments is not impaired. So while their memory strength can be weakened, you can still assess they can still assess it properly. It's not like that capability is gone.
So how could you tell that if the memory strength is weakened, so they're aware, but their confidence is high, although
well, the way to test to see, because there are, of course, other diseases, frontal lobe diseases or executive function diseases, where the correlation between your ability of saying hi and the memory actually being high is disrupted. But here, so the way we do this is we compare the accuracy. That's one of the behavioral slides I showed. The accuracy between when you say high and low, and see is the accuracy actually different. And you can see that by and large, this reflects reality, meaning they can do this. So the second part of your question about this being close to auras or before or after seizures, that is very critical. And we make sure that these experiments are done at least 4 or I think even 5 hours away from seizures. And if somebody is still in a post-ictal state, which sometimes can take much longer, we wouldn't do experiments then. So we will, and this is assessed by looking at interictal discharges to make sure that brain activity looks normal, because I think will clearly change.
Yeah. So Dr. Gaunt, the question is, have parallel studies been done for those with spinal cord injuries affecting only the lower extremities?
Yeah, so the question is, we're focusing on hands and arms. Have people looked at legs in terms of the ability to decode intention from the legs? And as far as I'm aware, this has really not been done with these types of devices in terms of single unit recordings in people. There's been some work in non-human primates looking at this, but really not in people. People have used ECOG style arrays to look at lower limb function, maybe in terms of control, but really the vast majority of the work cortically in terms of motor control is focused on arms and hands and much less on legs. I think one of the reasons for that particularly is that when you think about rehab or robotic strategies to improve function, there's a lot of interest in hands and arms. Locomotion or mobility issues are maybe reasonably well solved, although, certainly not completely, by wheelchairs. Maybe now with sort of the advances in lower limb exoskeletons for mobility, there would be increased interest in sort of extracting control signals from these parts of the brain. One of the challenges is that the legs are represented in the brain basically right along the midline and are quite deep. So actually physical access with the types of devices that we use would be much more challenging for legs than it is for arms and hands.
Dr. Buffalo, humans take about 15 trials to reach criterion, while monkeys take 30. Why do you think humans learn twice as fast as monkeys? And what type of monkeys are you using?
Thanks, these are good questions. So I neglected to mention the monkeys that we're using are rhesus macaque monkeys. And we choose them because they're really well characterized in terms of anatomy and physiology. They've been used in many studies. And their connectivity, particularly between the medial temporal lobe and the prefrontal areas that we're interested in, is very similar to human connectivity. And the anatomy of these structures are also pretty well conserved. So they make a great model for trying to look at interaction between these structures. In terms of why they take longer, so the task that I talked about, the Wisconsin card sorting task, is a pretty complex task. And we were actually thrilled to see that the monkeys could learn to perform the task, at a level that was fairly close to humans. Humans still have an advantage, though, because we can explain to them the rules of the game. So we can sort of, give them a head start in that sense. And also because humans have the ability, we think, to verbalize, internally verbalize the stimuli. That is, they get to kind of think inside, that it's going to be the blue stimulus and keep thinking of that, where that's not possible for the monkeys. We also think that humans are probably doing a better job at hypothesis testing. So keeping multiple potential target features in mind and then testing them systematically through the trials, which will get them to reach criterion faster. So we're working on analysis models that will be able to allow us to identify kind of the hypothesis testing that monkeys are doing and compare that to humans.
And you are doing this cross-species work, so what do you find are the most unexpected differences that you've found so far?
Interesting. Unexpected differences. I think, so for this task, actually, we talked about that the monkeys take longer to reach criterion for each new rule, learning each new rule. But there are other tasks that we've done where monkeys actually outperform the humans. And part of this has to do with, I think, the training regime that the monkeys are in, that they're highly motivated to kind of keep going and perform very quickly on the trials. And sometimes that can lead to actually faster learning than in the humans. So that's been kind of fun to watch in some of the experiments.
That reminds me of those Kyoto working memory experiments where the monkey remembers like 20 characters and their position on the screen, which a human could never do.
That's right. Right.
I think we see similar things, not in my lab, but in collaborators' labs that work with non-human primates, is that, the things that non-human primates and monkeys are good at and the things that people are good at are often just quite different. The real advantage of working with people, which Dr. Buffalo mentioned, is you can go in and you can explain the task, and 30 seconds later you can be doing experiments. Whereas, for monkeys, it may take weeks or longer to get that up. But, in our experience, asking a person to do the same task, over and over 10,000 times in a row is not a recipe for success. Whereas, in monkeys, such a thing is possible. So.
That's right. That's a great comment. Thank you. We have a question. I believe this is for Dr. Rutishauser. In regard to patients' confidence and decision time correlating with retrieval accuracy, as you mentioned, patients who did have high confidence in their memory actually were accurate. Do you then believe if someone simply had higher confidence and believed in their memory being good, even if it was not, their memory could potentially improve?
No, in that sense, that would be clearly a disservice, because somebody would believe that their memory is good when it in fact actually is not. And that clearly can happen, although that wouldn't be a classical memory disorder as such, but that would be an executive dysfunction. And some mental disorders clearly would reflect that, but I don't believe that would increase memory strength as such.
And speaking of which, we do have a question on neuropsychiatric disorders, so I'll just pose it to all of you. Can you discuss how a deeper understanding of cognitive control and abilities and then deficiencies in these functions could help lead to targeted therapies to treat neuropsychiatric disorders? So I guess how could your work extend to that? We'll start with you, Dr. Gaunt.
Sure. So, we often don't associate too many neuropsychiatric disorders sort of as being sort of exclusively within these sensory motor regions. And so our work is more focused on, on these control issues. But I think generally speaking, very broadly, the general idea that if we know the source of the error, if we know what circuits or regions of the brain are involved in this, then it gives us sort of therapeutic avenues to explore to treat disease states. And that's certainly been hugely beneficial in there's not a psychiatric disorder, but like in Parkinson's disease, where there's a location that we know that there's something's going on, we can then deliver stimulation to these areas. And there have been some recent some good work in major depressive disorders and other mood disorders related to deep brain stimulation. Again, where hypothesizing targets and places where things are going wrong can lead to then sort of the development of therapy for it.
Dr. Buffalo?
Yes, I'll just add to what Robert was saying. Some of that work on in both Parkinson's disease models as well as for treatment for major depressive disorders really highlights the importance of the circuit approach. So some of those therapies really work by, I think, amplifying or adjusting the connectivity between different brain regions. And that's something that we're hoping with this work is that we're doing as part of our U19 is that we can try to understand, again, using the circuit approach, what's going on in terms of memory formation. And through testing both in the non-human primate with mechanisms for modulation, either through electrical stimulation or pharmacological mechanisms, we can try to understand how we can affect that circuit, which then may be useful for future therapies.
So what I may add to this question is that one of the things that we have focused on in our grant that speaks to this question is on the role of different brain oscillations or in particular theta oscillations as a mechanism for cognitive control and memory ability. So what we are aiming for and what our hypothesis is that by understanding how theta oscillations facilitate coordination between the medial temporal lobe and in our case the medial frontal lobe during memory retrieval, that we then could design a targeted therapy that would either restore those theta oscillations to the way that we described it, or that would be in fact initiated in a closed loop manner at a particular phase of theta oscillation. So that would be one way by which we could facilitate the development of a targeted therapy.
And targeted therapies are so important because target selection is one of the hotly debated issues in, for example, deep brain stimulation. And that is relevant, of course, to these changes in synchrony for different disorders like the beta oscillations in Parkinson's, gamma in Tourette's, etc. And so being able to find these oscillatory dysfunctions and use them as biomarkers, and then use that to be able to really finely localize our targets, not just, for example, the STN, but what specific area of the STN and how much of it and when it should be stimulated. So I think that it's wonderful that we're moving in that direction. So we've just gotten started and unfortunately we're out of time. I'd like to thank all of you, Dr. Gaunt, Dr. Rutishauser, and Dr. Buffalo, for your time and for sharing your expertise. This was really wonderful. And thank you to our audience today for your thought-provoking questions. We can continue asking questions of our speakers, and they will be able to follow up with you via email. So thank you again to our speakers. And coming up next, actually speaking of circuit dynamics, please join me as I present on deep brain stimulation, opportunities, and ethical dilemmas. I hope to see you there.
Thank you.
Thank you.
Bye-bye.